Imaging cells is an integral part of the work of most labs around the world, and many of these labs run into the same issue. They're not able to image many types of cells for long periods of time without them starting to die. A cell thrives when stored at 37 degrees Celsius with 5% CO2 and 90% humidity in the atmosphere around it, which is why we store cells in an incubator such as this one. Cells need this environment to survive long-term imaging, but it is not economically or physically feasible to make the entire room where the imaging would be done mimic the environment of an incubator that could supply this. So, to get around this issue, we bring the environment to the immediate area of the incubator. This is often done with an incubator that can surround the microscope, such as this, or an incubator that fits on the stage of a microscope, such as this. We developed an on-stage incubator to allow the system to be portable and interchangeable between different types of microscopes, and we plan to be able to control the temperature, humidity, and CO2 levels within the chamber as it fits on top of the stage. It will be open to allow for several types of cell media holders to be placed inside. And now I will play a video explaining how our prototype works.
First, we'll place the environmental chamber on the microscope, then fill the small water reservoirs there with a little bit of water. We'll then connect a system of tubing from the inlet of the chamber to the outlet of the air pump, and the inlet of the air pump will feed through this water reservoir, through a system of tubing, and into the solenoid valve.
From the solenoid valve connected there, you can turn the system on, opening pressure going to the tank. This will insert CO2 into the humidity tank that was shown before. The humidity tank has water inside, which is heated by the blue plate underneath, and it is pumped out by the air pump at the top when the system is turned on. That is determined by the humidity, temperature, and CO2 levels, which are measured inside of the chamber of the cells for the humidity and temperature, and for CO2 inside of the reservoir there.
Once the gas system is set up, we'll plug the AC power adapter into the outlets that heat the blue plate, which heats the water reservoir, and to heat the heating system. So we'll plug this part into here, and this will send current to the heaters on the chamber,
As well as the solenoid valve controlling air inlet and the air pressure pump.
And once the system is all set up, you can open the Arduino program and click upload. Once it's fully uploaded, you just want to open the serial monitor to view the temperature, pressure, the current temperature. It'll prompt you to input the set point temperature, and it'll start measuring.
Now that you've seen how the device is operated, I can show you our results thus far. Here you can see the cells at the start of our testing process in 3 environments. From left to right, we have the cells in our prototype, cells left out in our lab, and cells that were placed in a full-size incubator. After leaving the cells in these environments for 5 hours, here are the results. The cells in our prototype are comparable to ones left inside of the incubator, while cells left at room temperature have started to swell, indicating the start of cell death. Further testing and iterations would be needed to determine if the cells would be able to last 24 hours in the system, but our current prototype shows plenty of promise.